Thank you. Thanks. I hope everyone's having a good time at SciPy. This is, I think, my favorite conference of the year now. I think PyCon has sort of gotten too big to really, I think, exchange ideas. I think also science has taken has sort of been superseded by web frameworks. So I kind of feel like this is the best conference of the year that I go to. Just to show of hands, who here is a biologist? Okay, great. Who here is a computational science or computer person that helps biologists? And who here is a domain scientist of another discipline? Okay, great, awesome, cool. Well, like I said, SciPy is pretty good. I don't know if anyone went to the reception last night, but I learned something new about scientific programmers. What's the difference between a scientific programmer who's shy versus an outgoing scientific programmer? An outgoing scientific programmer walks around looking at other people's shoes. So the purpose of this talk is to sort of educate you guys about what I feel are good ways of doing very large scalable genomic pipelines. In a lot of scientific workflows, especially genomics, it's not a model and sim game, and the bottleneck is really about moving the data, especially when you're talking about situations where you have large data sets and the institutions that are hosting and have those data sets or publishing those data sets are not the ones who have to actually get access to the data and analyze it. On the schedule, I'm supposed to give this talk with my colleague Jim DeLeo. Jim could not make it, so I'm giving this talk by myself. But he was 50% of this project, and he is the director of scientific computing at the NIH Clinical Center. So I'm not a biologist. I'm going to try my best to present the biological part of this talk in a way that may be high level to hide my ignorance, but not to misinform. So if there's anything that I totally screw up, please let me know. But yeah, I mean, we're basically, this is pretty a high level talk. I'm going to present a pipeline that we built, but the context is to try to show why we built that pipeline, not that pipeline is necessarily itself interesting, which I think it's interesting, but that's not the point. So the mission here is to empower citizen scientists to analyze many petabytes of public genomic data. And I say citizen scientists because we're not assuming that you work at the Mayo Clinic or NIH or an institution where you have access to data or you have access to resources, infrastructure that other people don't have. It's 2014. The cloud's been around for 7 years or so. So we assume that utility computing is an assumption. And at the same time, there's a lot of tools that are built around that to make it very easy for someone to build one of these pipelines. So we're gonna focus on that. And again, we are scientific programmers or domain scientists, so we really want to hide the plumbing, and we don't want to focus on the details. What we want to focus on is asking a question of the data, getting the results, and then moving on to another question. And then, so the algorithm I'll get to a couple minutes that we're using is a Secretary algorithm, but it's, again, the context is showing how I think you should build a genomic pipeline. So here's my overview of some of the biology terms we're going to use. So with next-gen sequences, you're generating a lot of data. And the sort of status quo of comparative genomics is really alignment. You're taking those images you get out the sequencer, you're inferring a nucleotide sequence, and then you're using that nucleotide sequence to basically figure out a diff of some reference genome. And that's what you get. A FASTA file is essentially the ATGC ASCII file. But the way that these large data sets are distributed and published is they create what's called a SAM, or a sequence alignment map, where you say, my genome and your genome are probably 99.999% the same. So why would we say, here's your full sequence and here's my full sequence. So instead, you're the reference, and then we say, well, Chris has these differences. And that's how we publish the data. And a BAM is just a binary SAM. So you create this sort of reference map, and then you just make it binary. And then the National Institutes of Health in Bethesda, Maryland, is sort of, you all know them. If you're working at a university or you are doing any kind of biology, you're probably getting funding from them. And then within the NIH is the National Library of Medicine and the National Center for Biotechnology and Information. And their job is to be the data warehouse for all of this genomic data. People submit data to them, and they publish it, and they make it available. And this is important because we're going to talk about how to get to that data. So what's the point of genomics? These are my kids. What's the point of genomics? The point is to improve health. The point is to advance fundamental science and cure diseases and improve clinical outcomes. And I think that's really important to focus on. If Jim was here, he would be yelling about that. That's his shtick. Is that it's not about saying, hey, we did something computationally interesting. It's about mapping that to clinical improvements. So here's a sequence alignment map. This is the 1,000 Genomes browser. Now, Jim and I are taking a different approach to comparative genomics, but I'm not saying that what we're doing with alignment is not great. Over the past 15 years or so, there's been a number of incredibly interesting tools developed to take alignments and understand that and try to map that to diseases and try to figure out the differences. If you go to the 1,000 Genome browser, you pick a gene, you pick a sequence on a chromosome, and it'll tell you all this interesting information about okay, well, African Americans tend to have this difference, and Utah residents have this this difference, and the Han Chinese have this other difference. The data is incredibly well curated, and they're really good tools for understanding that data. So we're kind of taking a step back and looking at what we're doing. So what is DNA? I mean, we all know how DNA works, but if you look at it, it's getting parsed, right? That's the whole point of DNA, is that it's, with protein synthesis, it's actually getting parsed and turned into stuff. And there's rules about that parser. So taking a step back and, looking at some of we've done some compiler designs projects in the past, and sort of looking at it that way, what does this remind you of? That's one of the original drawings of a Turing machine, right? So we're parsing DNA. DNA gets parsed the same way that code gets parsed. So what we're doing is we're basically going to say that the DNA can be expressed as a grammar. And this isn't a novel idea, but we're going to focus on it. In 1997, Craig Neville Manning was a graduate student in New Zealand and published a paper called Identifying Hierarchical Structure in Sequences, a linear time algorithm. It's a linear time and a linear space algorithm, which is important. So this is the Sequoia algorithm that we're going to use that was published in 97. What it does is it basically takes a sequence, a string, and creates a context-free grammar. It's not important for this talk, but a context-free grammar that that represents that sequence. So it sort of reverse engineers the rules of a grammar or language based upon the sequence. Resilient. I'm at Resilient, and we are doing this project, and we're sort of testing this project with NIH. But John Reel, who's my co-founder, is giving a talk tomorrow that's going to be focused on the specifics of the project. The project's called Web of Trails. It's open source. It's on GitHub. But he's going to focus on the specifics of the grammar and why we're doing it this way and some of the performance advantages. But we're just kind of taking this as an example for this talk. So I've been at SciPy, and there's been a bunch of talks about search and indexing. It seems like search and indexing technologies is a thing this year. And I've heard this repeated in multiple talks, where if you take a step back and you go to the theory, there's sort of different ways of indexing data. There's a hierarchy. There's lexical, syntactic, and semantic. where word count is lexical, and there's a lot of argument right now in some of these talks about what the best way to do syntactic analysis or syntactic indexing of data. And then semantic seems to be this dream that's sort of just over the horizon. So I'm applying this to genomics. I'm saying that what we're doing right now with alignment, alignment-based comparative genomics, is it's less lexical analysis. That's great. That genomics has been around for a couple decades tops, and so I think that's a good first approach. What we're trying to do is syntactic analysis, where you're actually creating a deeper understanding and a deeper a more in-depth representation of the data, and then trying to understand, well, how does that, what can I learn from that? Okay, so that's where we are in terms of the biology, but what I really want to get to is how do we build this pipeline? What are the best tools, what are the best Python tools for building this pipeline? And what do we see as a result? This is my favorite quote from college. But how do we get away from this, right? I mean, this is true. Everyone's done the math in classes. But how do we get away from this? How do we get to the point where we can just click buttons and data moves? So some of you probably work at national labs or large research universities. So Internet 2 is amazing. I mean, I've been using Internet 2 on a daily basis for 10 years, really. I used to work at national labs. So here's the whole thing, is that all these, in genomics, all the people that want to, all the people who own the data, who are publishing the data, who are creating data warehouses, they all sit on Internet 2. However, your NIH grant recipients, your small companies, your individuals, your citizen scientists don't have access to Internet 2, but actually they do. So this is a map. It shows the NIH main campus, and it shows Amazon's Amazon Region East data center. So it's 25 miles away from each other, and the fact that I think it's about 2 years old, but it's not well publicized, and it should be, is that Amazon and NIH are both peered on Internet 2. That means if you are Joe Public and you spin up a machine on Amazon's East region, you have a 100 gigabit connection to NIH, to the data NIH, and it's going over, it's 25 miles long, so it's, the speed of light is well under a millisecond, right? So now anybody who can use, a public cloud can now access that data, read from that data, read from these, 30 petabytes of data, as if it's in their own data center. And it's utility computing, so you have all the compute resources you want. So there's nothing to do, which is great. And again, I'm surprised this isn't publicized more. If you're on EC2 and you're talking to the FTP server, the data is just going over Internet 2. There's no routing. There's no special configuration. Okay, so that's how you get the data. That's how you can spin up a machine or spin up a cluster and grab data incredibly fast, as if it's in your own, top-notch data center. How do you do that without, writing a lot of code, right? So I don't know if anyone's here familiar with mrjob. Okay, so I saw 3 hands. So this is I'm glad I'm bringing this up. So mrjob, MapReduce job, it's essentially a Python project that abstracts a lot of the MapReduce details away. It's designed to run on, you can run it locally, you can build and test, local Hadoop jobs, which doesn't really make any sense, but it's good for testing. And you can also have that, interface with an existing Hadoop cluster, or it also interfaces with Amazon Elastic MapReduce. So here's, so we wrote, we've implemented Sequitur in Python. It's on GitHub. And so the very simple example where you're just taking a string and you're generating a context-free grammar, given that code, given that function, that method, to make that run on MapReduce, in like to treat elastic MapReduce as a giant batch cluster that you own, is pretty much one line of code. You import MR job, and then you create a class, and you just like I said, you just call your function and yield the result. So that's great. So you can now, if you have some Python program, some Python function you wrote, and you want to, run that on a petabyte of data, a petabyte of genomic data hosted on NIH, you can create that batch job in a line of code. Just feed it all the data you want to run against. Now, we didn't just do that. We also took Sequitur and we actually made a MapReduce Sequitur. So we're not just taking a genome and parsing that. We're actually taking the genome and splitting it up for MapReduce, and we can do that. And again, this is the actual grammar-specific code is on GitHub. But here's the MapReduce runner for that code. It's this is the version 2. This is the good example, and it's really not that hard. I mean, look, it's a few lines of code. You're basically just saying, call this, move this data around, split this split this bigger problem, these smaller problems, and that's it. So we ran this, and I want to be clear. This project is ongoing, but we ran this, and this is really what our pipeline looks like, and I've tried to be clear. It's pretty simple. All we did was write a MapReduce, write a simple Python program, and use MR job. And so here's the data. It's moving from NCPI to S3, and then it's running on MapReduce. Another thing you may be interested in is that MapReduce. The MapReduce service can now support things like Spark. You can search and find that out. It's not just Hadoop. We're doing this on Hadoop, but you can do much more complex jobs now. So we ran this, and without even trying to tune it, without even trying to really get better numbers. I have the. Oh, there it is. Okay. Just off the bat, we got 1/3 of what the theoretical max would be for the 100 gigabit pipeline. So this is the performance of a large stack of very fast SSDs, working together, aggregated together. So this is sort of the punchline. You write a very little bit about a code. You say, here's the data I want to analyze, and you now have you're now harnessing effectively a supercomputer, and the data is as if it's local to that supercomputer. And moving data from NIH to Amazon is free too. Moving data into Amazon is free. So it doesn't. This whole job took like $40 to run, to process probably about a few terabytes of data across many machines very quickly in parallel. So, so this is ongoing. Now that we sort of have a functional pipeline, and it runs pretty well, what we want to do is we want to take our method and do a cook-off against some other methods. Take some well-known phenotype, genotype data, and map data, compare alignment, compare hidden Markov models, and compare expert systems. Jim develops a lot of expert systems at NIH, and so he'd be able to build some specific to this problem to see what would be a better approach. In addition to that, one of the interesting things that we figured out about Sequitur after we started using it was that the data that you give it at the beginning affects the rules that are the rules of the grammar that are generated. So if you, for this example, if you start with saying, okay, here's some codons, here's some protein sequences, then the rest of the data it parses along those lines, right? In addition, NIH has a lot of electronic health record data from all the clinical trials, and there's not a real good approach for analyzing that data. A lot of that has to do with the fact that electronic health records are effectively new because no one's really adopted them until the American Affordable Care Act came out and incentivized that. So there's a lot of EHR data being generated, and we think that taking a sort of naive grammar-based approach, you can start identifying patterns in that data. Because that's really what this grammar-based approach is doing. It's taking a bunch of data and it's inferring what the patterns in the structure are. In addition, there's a lot of neuroimaging data. John, who's going to be talking about the WAT project tomorrow, is working on another project at NIH doing a lot of neuroimaging analysis on the cloud. And then this is why I love SciPy. So I didn't know about Scikit Bio until this afternoon with that talk. So it looks like a lot of their functionality I could leverage to replace some of the code I've had to sort of hackishly write to convert between different file formats. So I'm going to talk to those guys and see if I can use that to clean this code up, but also maybe there's some collaboration. That's it. I'd like to thank everyone that was involved in the project that we're talking about right now. And again, it's on GitHub. Jim DeLeo, who's the director of scientific computing at the NIH Clinical Center. Jonathan Simon, who's helped out. He works with the FDA. Ben Busby, who is sort of the bioinformatics liaison at NCBI, and then John Reel, who's also at Resilient. And again, John's talk is tomorrow at 3:30 if you're interested. It's going to go into specifics about why a grammar-based approach is a good idea for just data in general. And I'm on Twitter, and feel free to email me and check out the code, and I'd love to hear what you think about this. Nothing was supposed to be groundbreaking this talk. I just want to get the word out that this is how easy it is to build a petascale genomic pipeline. Thanks.